All right, thank you, Sarah. Thank you very much for the very kind invitation. And I mean, just to say I'm local, my office is PSD 201, literally less than 30 seconds by foot. So let's keep it a very informal, interact whenever you want. And if there are more discussions we want to have, please feel free to stop by my office or send me an email. I'm 100% hired by SMS, but I'm also at the same time a advisor, thesis faculty member of the Department of Physics. So actually some of the students are from physics are working with me. So I think this is a great platform and a great opportunity for me to also introduce myself, introduce my research in front of this particular audiences. So again, my name is Xie or Mo, that's easier. And today I'll be talking about the molecular biophysics from two domains. One is conventional NMR, the other is optically detected quantum sensing. And I will just start by saying that I am always amazed by how biology works. So if you try to say, look at a corner of the cell, you might see a plethora of different biomolecules. Each of them have their well-defined structures. We're talking about the proteins, DNAs, RNAs, lipids, small molecule metabolites. They all have their very defined chemical composition as well as molecular structures. These molecules also interact with each other. For example, here is the pathways of p53 mediated apoptosis of the cell. It's very complex. And these molecules are also moving. They're not static. They have motions on different domains from a very fast bond vibration to protein loop motions or domain motions in an enzyme. That's how they digest small molecules. And also protein folding, some of the protein will take forever to fold in the correct form, maybe with the participation of other molecules. Other proteins can fold in just less than a second. So in my opinion, if we really want to understand how biology works or what are behind cellular processes, it is very important that we go to the molecular level and have a mechanistic understanding of their structures, interactions, and dynamics. Of course, over years, people developed so many different experimental techniques to help us get information of this features or properties. And today, we're going to focus on spin magnetic resonance, or in particular, nuclear magnetic resonance. Let me start by giving a little primer. One-half spin systems play a very important role here. The reason being, if you apply a biased external magnetic field, these spins will somehow align to this magnetic field. And because they are one-half spins, you get two energy levels. This one-half spins include electronic spins or some of the organic molecules that has unpaired electrons. And also many nuclear spins, for example, hydrogen-1, carbon-13, nitrogen-15, fluorine-19, phosphorus-31, many of them are already very abundant at natural abundance in biological systems. So if we apply this magnetic field, we'll basically bias their orientation to have some sort of a polarization. So we can, by the end of the day, get a neg magnetic moment generated by these spins in the external magnetic field. So that's what the process one tells us. If you apply magnetic field, your spins will reorient and then align with your magnetic field. Then if you apply a radio frequency pulse with the frequency matches or resonantly matches the Larmor frequency of the magnetic moment, depending on the duration or the period you apply this pulse, you can basically nutate your magnetic moment within this Bloch sphere. Let's say if we just apply 1/4 of the time of the period, then we bring it from the z-axis to the equator. Now, once this magnetic moment is on the equator, it will naturally process along this or in this equator. So the precession is a kind of can be understood by making an analogy to classical physics. And the frequency of this precess is known as a Larmor frequency. Of course, we care about the nuclear spins in this particular colloquium, but just to mention electronic spins, we'll also have a gyromagnetic ratio, and we'll also do the same thing. The only difference is the gyromagnetic ratio of the electron is almost 4 orders of magnitude higher than that of nuclear spins. So after the second process, the nutation, you just use whatever detector you can have to measure the precision frequency, and you can record that frequency. And if you do a Fourier transform, you'll get a bunch of resonances. Now, anything that affects the precision frequency, even though just a tiny bit, let's say different bond connectivity or different density of electron clouds, or even some chemical processes or motions, that will change the Larmor frequency just by a little bit. That's why on the ppm level, you can see, even though they're all proton or hydrogen 1 spins, they process a little bit at different frequencies, and we can really resolve all of these different frequencies, and they can tell us a rich information about the molecule, their structures, their dynamics, and many other information. So in today's colloquium, again, let's keep it very informal. I will discuss 2 domains of magnetic resonance research done by both the conventional NMR spectroscopy. You might have seen these giant magnets in particular chemistry department, where almost all the research-based universities will have a NMR center or facility, as well as nanoscale NMR that is enabled by a diamond quantum sensor that is recently developed in the past 20 years or so. So here's the outline of my colloquium. In the conventional NMR spectroscopy section, I will give 2 examples to see how we use conventional NMR to study supra-tau dynamics in protein loops. And the second example will be related to intrinsically disordered proteins. I think it's really good to talk about this since Sarah is my host, and there are a lot of research interest in the physics department on IDPs. And in the second part, I will be giving some very general overview and some milestone works about the diamond quantum sensor-based NMR, as well as some of our rather comprehensive efforts to make this particular sensor more biocompatible. And it is followed by conclusion and outlook. So let me give a little bit of motivation why we want to study loop dynamics. So here in the blue part, that's the colosine E7. In particular, it's the DNA's domain of the colosine E7, which is a bacterial toxin. So some bacteria, they want to kill other bacteria in the environment so that they can win in the competition of resources. So they express this toxin, which is a protein. And other bacteria don't want to be killed, so they will express a immunoprotein, in particular, immunoprotein 7 or IN7, that counteract this toxin effect by tightly bound to the colosine E7 domain. And from a biophysics perspective, we're very interested in understanding what are the motions associated with these loops, and why and how they contribute to the tight binding of these two domains. Conventional NMR spectroscopy is a very mature and very powerful technique. Over years, people have already developed a large toolkit to measure different or events that happens on different time scales. And if you look closely at this chart, you will actually notice a gap, which is the nanosecond to microsecond time scale. So motions happening on this time scale is pretty difficult to measure spectroscopically. And it is actually easy to understand because in the solution, proteins are moving, and there are the tumbling motions and there are the translational motions of the proteins. So imagine if you have some internal motions or dynamics that happens on a slower time scale of the tumbling of your protein, then spectroscopically, you're just not going to observe them. Unless you freeze your protein, then you're not measuring anything in the biological relevant environment. Therefore, this supratulsine motions is actually a very well-known challenge to detect. Now, before going to more discussions in depth, let me briefly introduce how we do protein NMR, just to make sure everyone on the same page. So proteins are basically a biopolymer or a peptide chain that connects individual amino acid residues. So here I'm showing 3 different amino acid residues. And if you follow this backbone of the peptide chain, you will notice on each residue site, you have 1 proton chemically bonded to the nitrogen. So we know proton or hydrogen 1 natural abundance is 99.98%. So everything is basically hydrogen 1. For nitrogen, if you isotopically label them with nitrogen 15, then we have 2 types of 1/2 spins. You can do a multi-dimensional, in this case, two-dimensional NMR spectrum. This one in particular, it's a HSQC spectrum, or heteronuclear single quantum correlation spectrum, where you basically have the Larmor frequency, or the frequency of a hydrogen nuclei on 1 dimension, and the other dimension being the nitrogen 15 precision frequency. So basically, each peak on this correlation spectrum represents one of these proton nitrogen vectors. By doing some fancy tricks, in particular triple resonance assignments experiment, we know which peak belongs to which residue site, and that is, of course, important. And then by the end of the day, by analyzing or study the spectral changes of each residue site or each peak, we learn about something of the particular residue site. For example, we care about the T2 or transverse relaxation rate. Intuitively, if you have a protein or a domain like this, for the flexible loop, because they're moving very fast, you get more motional averaging, and that leads to a sharper peak or narrower line width or slower relaxation rates. Conversely, for the structured domain, compared to the loop, they move slightly slower, and that gives you line broadening effect. So basically, you lose your coherence faster. Now, by analyzing, let's say, the line width of each peak, you learn about the mobility or flexibility of the protein. Now, if we go back to our research question, can we somehow look at supratulsine motions that are slower than the tumbling rate of the protein? How about we just slow down the protein rotation rate or tumbling rate in solution. So to do that, we basically add some silica nanoparticles that are 20 nanometers in diameter compared to protein molecules. The nanoparticles tumbles much slower, and the protein molecules will occasionally bound to the surface of the nanoparticle, effectively slow down their tumbling rate, and then got released. So it is really this transient binding and also fractional binding that makes this method work. Let's take a look at the data. What do we get here? So in this plot, I'm showing S squared, which is the order parameter of a protein. So it's a normalized number between 0 and 1. 0 being you have a region that is very, very mobile. There's no way of any sort of a correlation. And 1 being this site is completely rigid. So it has no relative motion compared to the global motion of the protein. So normally you get a number between zeros and ones. The x-axis is the residue number. So if you have a protein, you stretch it and make it a linear chain, and you count number 1, number 2, number 3, number 4. So that's our sequence or primary sequence of the protein molecule. Now, if you look at the data, let's look at the blue curve first. You'll see this is the linear form of IM7 protein, which is our model protein here. You see some flexibility in the N-terminus, and then you have a few helix regions that has high order parameter, and then you have this loop 1 that has more mobility, and then rigid region, mobile, rigid, and finally you have a mobile tail. So the blue trace is the so-called model-free analysis, which is let's say a data interpretation method for traditional or conventional relaxation experiments. So you collect the R1 relaxation, R2 relaxation parameters, as well as a heteronuclear NOE, which is a nuclear Overhauser effect. So all of these relaxation parameters, and you fit into the model-free model, it outputs some parameters, tells you what's the tumbling rate, what are the other parameters for individual site, as well as the correlation time for each individual site. Again, by the end of the day, you get this trace that reflects how many motions are associated with each site. Now, the red data represents the nanoparticle edited version of the spin relaxation. Now, so it largely follows the same trend, except for the loop 1 region. You actually see more mobility on the S square parameter, order parameter. And this is really the detection of the supra-Taussig motions within this loop, because the conventional or traditional model-free analysis or spin relaxation experiment is not going to capture these very slow motions that are slower than the tumbling rate of the protein. So you simply don't see them. But now, once we effectively slow down the tumbling rate of the proteins by adding nanoparticles, we open up the window and we can really see the additional supra-Taussig or nanosecond to microsecond motions. And I mean, NMR, a good thing about NMR is that the theory part is very rigorous. We can basically get the analytical solution to explain this phenomena. So this is also very important from a molecular dynamics simulation perspective. So we compared our data to MD simulation. So first of all, not all the force fields works. Many force fields does not. I'll tell you why. And then secondly is that so these are the MD traces by applying different averaging windows. So we ran the trajectory for 2 microseconds. You really see the dipping or kind of corroborated or consistent results when you apply a MD trajectory averaging model that is on the microsecond time scale. So in other words, if you just use traditional model-free analysis, your detection of the window is restricted on this kind of single-digit nanosecond time scale, which is the tau c of the protein. So you only dip as much as here, but only when you do this additional motion averaging, you can capture what we observed experimentally. So this actually provides some experimental data to benchmark the force field development, because most of the force field are developed 10, 20 years ago using, back then the computational power was not long enough. You barely get any trajectories that are longer than 5 nanoseconds. So you can only benchmark your force field to that time scale. Now we provide direct experimental validation to help refining the protein force fields. So just to briefly mention, we're not going to too much into the biology, but we have used this NASAR nanoparticle assisted spin relaxation method to study KRAS protein, which are very important in kind of regulatory tools. And if these proteins misbehave, there's a higher risk of association to cancer. So we are basically able to see additional nanosecond to microsecond motions in some of the loops and try to understand what are the conversions between their inactive form and the active forms, and then also the behavior of the mutations. And it's very important to point out that if you don't use NASAR, which are basically these black data traces, they're literally flat. So there are no much motion on the time scale faster than nanoseconds, but there are slower super tau c motions. So let me pause for a second and give a quick note. What I have introduced is basically we extend the conventional spin relaxation observation window by using nanoparticle assisted spin relaxation, and then open up a observation window all the way extend to the millisecond region, and thereby we close this gap. Also did a careful theoretical understanding, both within the red field theory regime, as well as using stochastic Liouville equation to see how chemical exchange happens on these time scales. So for those of you who are interested, there are some papers from my colleague, Greg Jameson. Now, for those of you who are thinking very critically, I said, when you have a protein molecule that comes interact with the nanoparticle surface, will there be any preferential binding orientations? Will there be any binding that affects the natural behavior of the loops? So we also ask ourselves the same question. Now let's take a look at the data closely. So the nanoparticle assisted spin relaxation basically takes the difference of the T2 measurement with and without nanoparticles. And if you look at the raw data before you do the subtraction or take the difference, you actually see a lot of sites that has very high R2 rate. Those are associated with chemical exchanges. And once you do the subtraction, those effects actually nicely cancel out, meaning chemical exchange are not likely to be affected by nanoparticle addition. However, if we use different types of nanoparticles, some of them are negatively charged on surface, others are positively charged on surface, you actually see slightly moderation on the S square order parameter profile. So both ourselves observe this as well as some other researchers using our nether methods. They observe that, and so we're very interested about it. So it seems that disordered region will have some sort of interactions with the nanoparticles. So we say, why don't we just look at IDPs? These are completely disordered proteins. They comprises of over 30% of the eukaryotic proteome, and they have very important biological functions. From biophysics perspective, if you think of their free energy conformational landscape, they actually, the energy landscape is very flat, meaning compared to a structured protein, you have this very narrow landscape. Your protein will just stay in the lowest energy form. If it wants to change from one conformation to a different conformation, it has to pay a large energy penalty. But for IDPs, it can just randomly or freely change the conformation without paying too much energy penalty. So structurally, they're very flexible. They're typically very soluble. Effectively, each site will have the equal probability to interact with the environment, or in this case, the surface of a nanoparticle. So we chose p53 TAD, which is a transactivation domain of the tumor suppressor p53, which is the IDP, as our model system. So here, basically, it's the same thing. We look at the delta R2, the R2 measurement with or without nanoparticles. We take the difference. So basically, this profile plotted against the residue number tells us each segment whether it has a stronger binding tendency to the nanoparticle surface or not. So just to point out, here we're dealing with a very weak binding regime. We're talking about KD in the millimolar region, because if you have a protein that tightly binds to the nanoparticle surface, the tumbling is so slow, you wouldn't even see any NMR resonances. So here it's very weak binding, but we see these fine features of preferential binding in each segment of the protein. Now, if you really look at the sequence of the p53 TAD, you notice here's a positively charged lysine. Here are two positively charged arginine residues. We use silica nanoparticles with a pristine surface, which is slightly negatively charged. So yeah, it makes sense. Charge-charge interaction will likely to play a role. And we'll also have a bunch of negatively charged residues. So charge-charge repulsion are likely to produce these basins. We want to understand more than that. So we did mutagenesis. For those of you who don't know, basically during the protein expression process, you can do some tricks known as a site-directed mutagenesis to replace certain residues with other residues. So wild type is our reference. If we replace the proline 27 with something else, you see a drop in the binding profile. And you can isolate or do a subtraction of these two profiles to get the net effect of this particular proline residue contributing to the overall binding across different residue sites. And you can do that for negatively charged aspartate residues, as well as positively charged lysine 24 residues. And you see these net effects for each residues. We can, of course, model them with your favorite equation. So we use a symmetric exponential function to just try to capture these profiles. And we need two parameters to describe each profile. So basically, we need a lambda, which is how wide it extends. So it seems that the charge effect has really long range effect compared to the proline residue. And also we need this AJ, which is basically how high it can go. And for that, we did the same trick, but in this case, we just measure the 20 amino acid residues molecules in solution. So they're not proteins, they're just the protein building blocks, amino acid residues. So when we do that, we look at the relaxation rate of carbon alpha, and we get the AJ parameter. So now we basically have a way to describe each residue site, what is the contribution to the overall binding, and then imagine if we will sum up all of these contributions, hopefully we can recapture what the overall experimental data tells us. So for example, this is our data, and this is the back calculation with the model. We name it the free residue interaction model, or FRIM. And of course, we're not satisfied with one particular case. We try to measure some other IDPs with different lengths. By large, it captures the binding profile very rigorously or very quantitatively. And for those who are more critical, they're saying, this is not so good, you have this. And actually what is hidden here is that for each of the plot, we have to apply another global scaling factor, because FRIM does not necessarily take care of that. So we realized what is missing in the FRIM model is that here we're doing simple additions. So it is a linear additive model. What is missing behind is the cooperativity. So if you have two strong binding sites, the joint effects will be larger than the single effect times 2. So that's a cooperativity. Without going to too much derivation, we basically develop another model known as a simultaneous linker ligand cooperative binding model, or SILK model. In this model, we're basically, instead of doing addition, it is multiplicative. And this model are able to capture some of this net effects profile much better. And we also experimentally verify the existence of cooperativity by doing more fancy tricks with the mutagenesis. Now, if you look at the fitting to the global models of these 4 different protein or 4 different IDPs, it not only captures overall landscape, but also takes care of the differences in terms of overall binding strength. So of course, playing with the data fitting or modeling is always fun, but also it has some implications. So we're thinking IDPs are very abundant in biological systems, and they are actually very related to neurodegenerative diseases. Currently, there's no way to target proteins or IDPs as a drug, because for a small molecule, you can find the hydrophobic core and put the small molecule that binds it or inactivate the protein function. For IDPs, they're very flexible. How about we use nanoparticles to kind of design and engineer the surface so that by having some multivariant binding models, we can have some selectivity to target or drug IDPs. So people have published some perspectives on that regards. We're not experts in nanoparticle engineering, but we hope that this model or this type of analysis can help us provide or understand those processes. Now, let me quickly summarize. So conventional NMR has a lot of unique advantages. I'm not going to go through all of them, but it is important to point out that it does not require non-native labeling, like a fluorescent label. If you have a protein or if you have a molecule, most likely you will have some proteins or hydrogens and carbons, and we can just use their isotopes. And in many cases, you don't even need the isotope. They're already 1/2 spins. I discussed the 2 domains of proteins, structured proteins versus IDPs. But in real biological systems, there is a continuum. You can have very folded proteins. You can have complete randomness, but you can also have something in between. So it's at this stage, it seems that for the first part, the global binding, the overall binding affinity is stronger than the IDP part. So IDP interaction is really weak. That's why we can still, by large, trust the Nassar method. And also by using different nanoparticles with different surfaces can help us rule out those effects. But if we have a way to study some of these intermediate states that might help us to understand protein folding processes, for example. And also, I want to point out to some factual things. It's a great method, but we also pay a high price. We need to use a very high magnetic field strength. Some of them are 1.2 gigahertz, that's 28.2 Tesla. Very homogeneous. Many people don't realize that, but in order to separate the ppb level of alarm frequency procession, you need to make sure your magnetic field is very, very homogeneous. Therefore, there's a lot of coils that are just making sure that you produce a very homogeneous magnetic field. So very expensive. And also, it typically requires milligrams of sample. And the reason for that is because the detection we're using is a classical sensor. It's basically an inductive detector. When you have a rotating magnetic moment, if you have a metal coil, that will generate a current, and you measure the current. So what if, let's say, we're sample limited? You want to study the single molecule metabolism, or you want to study something that you can only extract so much from, let's say, a real patient from a tissue sample. Conventional NMR would not work. We want to see what we can do to improve that. So that brings me to the second part, meaning doing nanoscaled NMR using a quantum sensor. So I want to also give some very general introduction. We start by talking about the second quantum revolution. This semester, actually, the physics colloquia have already invited many researchers in the quantum technology research field. So quantum technologies, or basically, if you think almost exact 100 years ago, the quantum mechanics were formulated. In the past few decades, people keep working on this technology, and now we're in an era that we can actually engineer the state of a matter and then use them as very useful tools to look at the new scientific territories. And also, it's worthwhile to mention the Nobel Prize in Physics 2022 was awarded to 3 QIS scientists. I can give even 2 more examples about the quantum sensing in particular, which is one domain of quantum technology. So quantum sensing, here we just use a very generic definition, describes the use of a quantum system, of quantum properties, quantum phenomena to perform a measurement of physical quantity. So the first example is a LIGO, light infrared or laser interferometer gravitational wave observatory. So basically we built very giant arms perpendicular to each other, 4 kilometers each. And then we have a interferometer on these 2 arms. And then when gravitational wave comes from the universe, it stretches the land kind of slightly, and we measure the distance and in order to interpret the gravitational wave and hopefully use that to learn about the universe. And if you think of the error level, it already down to the 10 to the 23rd kind of a baseline, which means it can measure a distance change that is only 1/10,000 the width of a proton. So it's kind of crazy. My mind cannot process that information. And the second example, so this is on the kind of dimensional or spatial domain. The second example is on the time domain. People build atomic clocks. So basically, there are some atoms. They have a finite or kind of a constant transition frequency. And we want to build a device to measure those frequencies. The better you measure those frequencies, the better you can do timekeeping, because we just assume that these are constants, and we try to measure those constants. The better you measure, the better you can do timekeeping. So for atomic clock, it kind of keep going down. I think the latest results is that we have an error, relative error, that is on the 10 to the -19 level. That means if there is a stable atomic clock to measure time from the birth of the Earth, which is 4.6 billion years old, if we measure that duration, the error will be less than 0.1 second. Of course, for NMR, we care about a quantum sensor that can measure magnetic field. So that's why we want to introduce this diamond quantum sensor. Everyone is familiar with diamond, and sometimes if you go to a jewelry store, they'll tell you, look, we have a pink diamond. So one of the reasons to make a diamond pink is because of the presence of so-called nitrogen vacancy centers. So we all know the structure of a diamond. If you remove 2 carbon atoms and put 1 nitrogen atom back in, leaving the other side empty, you got a NV center. And that NV center is fluorescent. So you can really optically address each individual NV center. Now, in order to use such a defect as a quantum sensor, here's what we do. So this is one of the setups we built at the University of Chicago. It's basically a microscope with some additional functionality. Of course, we start with a piece of a single crystal diamond. By the way, all the naturally occurring diamonds are too dirty. They have so many defects. So we actually use a single crystal diamond, CVD grown. And we somehow manufacture the NV centers inside. Now we need to build a confocal microscope that allows us to look at each individual defect. And here is really the trick. So this is the energy level of the NV centers. It has a ground triplet state. Specifically, it has a MS equals to 0 substate and 2 energetically degenerated ±1 substates. If you shine a green laser, you will excite them. So 0 goes to 0 and goes back to 0 by emitting 1 photon. +-1 goes to +-1, also goes back by emitting 1 photon, but it also has 30% of probability to go through a singlet state, reverse the spin state, and comes back to 0. So that leads to 2 important properties. One, we can optically pump and initialize this state. If you don't do anything, you have a probability of a 1/3, 1/3, 1/3, and that's just a Boltzmann distribution. If you shine a green laser, let's say for a certain duration, 3 microseconds, for example, by the end of that laser pulse, you will initialize your state all to 0 state. So the probability will be 1, 0, 0. And the second property is that if it starts with 0, you go to 0 and it goes back to 0. So it always emits 1 photon. But if you start with ±1, it goes up and you will lose some photon. So that will be a dark state. So by looking at the fluorescence intensity, you know where your quantum state was. In practice, we also add a biased magnetic field. This is helpful because now, instead of dealing with this ±1 energetically degeneracy, we have 3 discrete energy systems. Now you can use 2 of the 3 energy systems as a quantum bit or qubit. You can use microwaves to drive the transition between these substates. And you can even prepare the superposition of this quantum state. And that makes it a solid state qubit that is very sensitive to magnetic field, because magnetic field will basically split these 2 energy levels. If you keep measuring the frequency of this, you can deduce what happens to the magnetic field along it or outside in the environment. So let me pause for a second to give a quick note. So the way to think about it is that the NV center is an artificial atom that is trapped in diamond lattice, pretty much similar to a trapped ion or trapped atom in vacuum, but now we're dealing with diamond. It's very stable. You don't have to worry about the vacuum leakage. It has very well-defined quantum states. It has very well-defined transition frequencies. Again, it's diamond, very stable. So, the way we clean diamond is that we use a mixture of perchloric acid, sulfuric acid, and nitric acid and boil it for 2 hours. So your diamond will be fine. Everything else will be destroyed. Very long coherence. We're talking about microsecond T2, millisecond T1, has magnetic field sensitivity. I mean, these numbers keep updating. And we do optical readout. So instead of measuring a current, we look at how many photons. And most importantly, it works at room temperature. Of course, if you go to cryogenic temperature, 4K or millikelvin, these numbers or these properties will be even better. But even at room temperature, it works. So now we have this magnetic sensor. How do we do nanoscale NMR? So here is a illustration. You have a single NV here, and you put your sample outside of a diamond. And effectively, this nuclear spin will process, and it will generate an AC magnetic field. So if we can measure that AC magnetic field using diamond sensor, we can basically do NMR. So for those of the expert in the audiences, this is basically the dynamical decoupling pulses in traditional NMR experiment to just apply a bunch of pipe pulses, varying the delay in between. And that frequency, if that matches with your AC magnetic field, which is your signal, you can register a resonance in real lab. This is basically the NMR signal we measured on objective oil. We always use it as a benchmark. So where is this technology standing at? You can use single NV center to measure very small volumes. You can also use many NV centers to get better sensitivity at a sacrifice of spatial resolution. It's all relative. We're still talking about the picoliter volume. Now, here are some milestone works. People have shown that using this configuration, you can measure the carbon-13 NMR signal from one single ubiquity molecule. This is because the sensing volume is so tight, it can fit in one molecule at a time. So you can basically do single molecule NMR. Let me say it in a different way. You have a molecule or a protein that has 200, 300 carbon atoms. And these atoms, or the nucleus of these atoms, generate a tiny magnetic field. And that tiny magnetic field can be measured by the diamond quantum sensor. Using this kind of ensemble measurement, people have achieved spectral resolution that is able to distinguish the chemical shift for this particular sample. And you can even do conventional NMR tricks by going to 2-dimensional or multi-dimensional NMR. I'm a little bit running out of time. I want to really leave some time for discussion and Q&As, so I'll speed up a little. So what is missing right now is that, again, this is a great work, but if you think about how the proof of demonstration experiment was done, you drop cast protein solution on diamond surface, you dry them up, you submerge your diamond upside down in objective oil, and then you shine laser over the sample for a couple of hours. So by the end of the day, you're probably only looking at some carbon atoms, no longer proteins. So it is very important to make the surface of diamond biocompatible and also build some flow channel for sample controlling. I can talk more about why we need to immobilize the sample on the surface. For the sake of time, I'll skip this. Basically, we developed a chemical way of functionalizing diamond surface. So basically, we deposit some thin layer of oxides, and then instead of directly functionalize diamond, which is chemically inert, we functionalize the oxides. And of course, we can control the density of the biomolecules of interest on the surface. We can use aptamers to recruit proteins or pull down proteins from a real biological sample on the diamond surface. And this biological functionalization does not really impact the coherence of the NV center, which is very important. You don't want any change in the surface, on the surface, to negatively impact the sensor performance. And just to say, ongoing project, we actually noticed that TiO2 is better material than alumina. It's more stable, and it can actually increase the coherence of NV centers. So research highlighted user title, quantum sensing goes bio. That's really what we want to do. And also another part, very important, we're dealing with diamond, and diamond is a very, very interesting material. It's very hard, so it's very hard to kind of manipulate or fabricate. But regardless, we are able to make very thin diamond membranes that is only tens to hundreds of nanometers in thickness. And if you look at the cross-section of this sample, we basically have a sapphire substrates, and then this is our diamond membrane. You can really see individual carbon atoms, and the crystallinity is just extraordinary. You go from a bulk property right into a bulk diamond surface. There's no much amorphous carbon. And that is important because for sensing, we want to use shallow envision centers, and you want to make sure that layer maintains good single crystallinity. And so here in the physics department, of course, I would love to work with Professor Bob and manage to keep advancing this technology. And once you have a diamond membrane, you can easily make a flow channel by bonding your diamond membrane on a quartz cover slip. And now because it's so thin, you can even do a turf illumination. We can incubate a cell on top. And moving forward, our idea is if we want to study this part of the cell or even this particular molecule of the cell, you can just find the NV right underneath to do the detection. Just quickly say the diamond membrane platform has many other potential applications. My colleague Xinhan Guo has shown that the diamond membrane, it's easier to engineer string within the material, so that can help us understand some fundamental physics or some hyperfine interactions on some of the group 4 vacancy centers in diamond. And also with Mark Kolonka at Harvard or MIT University, they were able to make photonic crystal cavities that achieve the highest Q with this diamond membrane. It's just because the fabrication method is much simpler now. So I'll skip the conclusion and just to say, my vision of this field is that the conventional technology will always bring us new knowledge. Quantum sensing represents another kind of a fast growing technology that can potentially allow us to learn many things faster and better. If you think of a ubiquitin NMR, that's what you can see with the conventional NMR, very nice splitting of the peaks. And here is the 2016 how it looks on a single molecule level. But again, it's a single molecule ubiquity. If you think what was some of the first NMR spectrum look like, that was back in 1951, 50, 60 years later. We don't know what this technology will be standing at. So my lab here, or my program here, will be focusing on many of the aspects of this technology, and to kind of study single molecule biophysics, as well as making some hybrid channels. I'm also very interested in CXFlow. I want to use the technology or this facility to see if we can understand the diamond material slightly better. I mean, I'm not an expert. I'm open to discussions and the potential collaborations, because I think using the fast X-ray pulses, we might be able to look at some charge transfer properties of the diamond material, which is very, very important to advancing this technology. With that, I want to thank you everyone for your attention and for the hospitality. And just to say, I'm already slowly assembling my team. I have 3 lab members, I mean, Vinod is still in India, a postdoc will come in May, and hopefully we'll work together and to make some progress together with the physics folks. Thank you very much.